Hello, my name is Mohammad Usman, and I shall be giving you a video presentation of the research article titled AFP LSE Antifreeze Protein Prediction Using Latent Space Encoding of Composition of Case-Based Amino Acids. Before starting, I would like to give you a brief introduction of antifreeze proteins. Well, it all started in the 1950s when Dr. Backus from the Cornell University asked a simple question to his senior colleague Dr. Scholender that when Arctic fish swim in the ice water at minus 1.7 degrees, why do not they freeze? Well, this simple question led to a series of research work that is still in progress. Dr. Scholender investigated and found out that some marine fishes living in the high north had a compound in their blood that actually lowered the freezing temperature of the whole fish, and he called this substance as antifreeze. The true nature of this antifreeze compound, however, was identified as proteins by Dr. DeVries in the 1970s while he was working with the fish species in the Antarctic. I discussed earlier that the antifreeze proteins are responsible for depressing the freezing point of the water in the species. They do so by performing two mechanisms. First is the thermal hysteresis, which is a mechanism in which AFP lowers the freezing point of the water without altering the melting point. Thus, it creates a gap between the freezing point and the melting point. And this process is termed as thermal hysteresis. Also, we know that ice has an unusual property of recrystallization, in which small ice crystals bind to the water molecules and become large ice lattice. This in turn damages the inner cells and tissues of the organisms. So AFP inhibits this process of recrystallization by binding to the basal plane of the ice, as can be seen in this figure. This way, the geometry of ice is changed and it forms a cone structure which has lesser edges, and it does not harm the cell. The antifreeze proteins are found to have numerous applications ranging from food products to human organ preservation. In the food industry, AFPs are mostly utilized in ice creams to maintain its shape, its appearance, and its taste when it's frozen or during the thawing process. AFPs are used in agriculture to plant in places which suffer harsh cold weather. Japanese radish is found to have AFPs in them, which is cultivated in winter season. AFPs are being studied to be utilized in cryopreservation, and as we are aware that every 10 minutes a patient is added onto the organ transplant list, however 50% of the donated organs die out before being transplanted to the patients, as they have only 8 hours of life. So AFPs combined with the revolutionary techniques can help to lead the way towards future where no patient must die waiting. Let's have a look towards the challenges involved in the prediction of antifreeze proteins. First, it is difficult to distinguish them from other types of proteins. Some AFPs are observed to change their structure at low temperatures, and the way they interact with ice differ from species to species. This may be due to the difference in the geometric arrangement of oxygen atoms in ice in different species. And finally, the AFPs of even closely related species lack in similarity and in structure and in sequence. To address this problem, many methods have been proposed. And with the advent of machine learning algorithms in the last decade, many methods are machine learning based algorithms. However, the pioneer work of proposing a machine learning algorithm for the AFP prediction is by Kandan-Swamy, who named his algorithm as AFP-PRED. This work was based on random forest classifier. Apart from random forest classifier, support vector machines have been utilized in a variety of researchers who proposed methods named as IAFP, AFP-PSSM, AFP-PSEAAC, AFP-COOL, and the latest work in this direction has been done by Shudat Khan, who proposed RAFP-PRED using the random forest approach. There are two vital steps to successfully address the prediction problem. First is to develop a significant feature set, and then to select an appropriate machine learning algorithm. First, I will discuss with the formation of feature set. To address the prediction using machine learning approaches, one must select a feature selection scheme and an appropriate classification method. For feature extraction, composition of K-spaced amino acid pairs scheme is used. This scheme is based on the frequency of amino acid pairs that are separated by K numbers of residues. This encoding scheme has emphasized the significance of amino acid pairs and have been utilized in various classification methods. This is the illustration of CKSAAP descriptor for K is equal to 2. If K is equal to 2, K-spaced amino acid pairs for J is equal to 0, 1, and 2 are considered. For each J, the corresponding features F0, F1, and F2 are computed. Each feature has length of 400, and the final feature vector is obtained by concatenating F0, F1, and F2, which has the length equal to 400 into 3. The value of each descriptor is calculated by dividing the number of occurrences of that amino acid pair by the total number J-spaced amino acid pairs in the proteins. In our previous research, we performed thorough experiments to determine the optimal value of K, and the best performance of the classifier was obtained by maintaining a gap value of 8. Therefore, in this study, we use the same gap value. While keeping the gap value of 8 in CKSAAP, the retrieved feature vector has a length of 3600. But training the classifier on less samples with large dimensions may result in loss of generalization for the new samples. Therefore, we intend to achieve a satisfactory prediction using reduced feature vector. The dimension reduction can be carried out using several techniques, including principal component analysis, Youden Index, and mutual information, etc. However, in this study, we opt to use autoencoder, which has recently emerged as a successful neural network framework that learns the input data in much fewer dimensions and then regenerates the output, which is approximately similar to the input. We utilize this property of autoencoder for the purpose of feature extraction in this study. We propose to use a multi-layer autoencoder architecture that has been regularized to be sparse to generate compressed latent space. By imposing this sparsity penalty during the training, the model learns the most discriminative features for AFP classification from the input data as a byproduct. This is the design of the novel autoencoder-based classification model for the prediction of AFP. The model is a combination of autoencoder and a classifier. As described earlier, the simultaneous training of autoencoder and classifier, a noise-free latent space is learned by the latent space which has least redundant and most relevant attributes of the input data. The architecture is composed of 3 sections: an encoder with some hidden layers, a latent space, and a decoder that regenerates the input from the latent space variables. The latent space here serves as an input for the classifier as well. The classifier, as you can see, is a fully connected 4-layered multi-layered perceptron, and it is tuned to perform prediction task. The model is trained using Python on Keras for 1000 epochs with a variant of gradient descent algorithm called RMS-prop. Each layer uses ReLU activation function except for the output layer of the classifier, which uses softmax function. Furthermore, a dropout layer of 30% is used after each layer for better generalization and to avoid overfitting. Look on the detailed network specification. The autoencoder has 5 layers, including the input layer and 4 hidden layers. The input of the number of neurons in the input layer of the encoder is equal to the length of attribute vector, which for in case of CKSAAP k is equal to 8 is 3600. The number of neurons in the first hidden layer is 50. The number of neurons in the second and the third layer of the encoder is 25 each, and the fourth hidden layer has 10 neurons. Decoder is a complement of encoder. This symmetry ensures the smooth encoding and decoding procedure. The number of neurons, therefore, in the first hidden layer of the decoder is equal to the last layer of the encoder and so on. The number of neurons in the first, second, and third and fourth layer of the decoder are 10, 25, 25, and 50 respectively. Finally, the number of neurons in the output layer of the decoder is equal to the length of attribute vector. The classifier, as I said earlier, is a multi-layer perceptron implemented to perform prediction tasks from the latent space variable. It is composed of 3 hidden layers and an output layer, and its input layer is the latent space. In the proposed study, the latent space has 4 neurons. Therefore, we can say that the input of the classifier has 4 neurons and each hidden layer has 10 neurons. The number of neurons in the output layer is equal to the number of classes, which in this case is 2. To decide how many neurons must be there in the latent space, we perform an ablation study. The purpose of ablation study was to obtain a simple overall architecture. We observed the effect of varying the number of neurons in the latent space and also the behavior of model being trained on variable data distribution. Figure A illustrates the workflow of the proposed method, in which features of the protein sequences are extracted using CKSAAP technique with k is equal to 8. Further, the training and test datasets are formed, which are evaluated using AFP LSE model, and prediction results are obtained. Figure B summarizes this process of ablation studies. As mentioned earlier, we observed the behavior of our model on varying the number of samples in the training and testing datasets, as well as the performance of the model varying on the number of neurons in the latent space. The features are extracted from the protein sequences using CKSAAP method, keeping the gap value of 8. Then a balanced dataset is formed having equal ratios of positive and negative samples, and the model is first trained by keeping 25 neurons in the latent space. After evaluation, the number of neurons are systematically reduced. Subsequently, an imbalanced dataset is obtained with 2 and 3 times more negative samples than the positive ones, and then same ablation study is carried out on the latent space variables. For each configuration, we perform 20 simulation runs, and each training dataset was divided into 2 subsets of training and validation with a ratio of 90:10. We used early stopping for the metric of validation loss, and the training was stopped at approximately 700 epochs. For the purpose of performance evaluation, we used widely used threshold-dependent biostatics nomenclatures, including sensitivity, specificity, and accuracy. The accuracy measures the correct prediction over all the classes. Therefore, in case of highly imbalanced data, if a classifier predicts only the negative samples, negative classes, the accuracy measure will still be very high. Therefore, in our study, we consider the Youden index value as the performance measure, which is dependent on both sensitivity and specificity. Here, figure 4 illustrates the values of reconstruction error at the decoder, and figure 5 shows the effect of Youden's index by varying the number of neurons in the latent space. The proposed model generates two types of outputs: a decoded feature vector and a class label of input protein. We used different loss functions to minimize their respective error values. The performance of the proposed classifier is compared with existing methods. This study evaluates the performance of the classifier on the parameters reflecting the true efficacy of the classifier by considering the imbalance condition of the training and testing datasets. Therefore, we emphasize on the parameters of MCC, balance accuracy, and Youden Index. It can be seen from the results that the proposed method achieved superior performance on all statistical measures. We also performed the screening of novel AFP candidate proteins obtained as an independent dataset containing 10 candidate AFPs from Interpro database. The proposed method predicted 9 proteins as AFPs and only 1 protein was predicted as non-AFP. Compared with the AFP LSE, PSI BLAST retrieved only 4 out of 10 candidate sequences as AFPs, whereas the NCBI database annotated 4 out of 10 sequences as hypothetical or unnamed proteins. Further, 3 of them were characterized as type 1 antifreeze protein or AFP-like domain-containing proteins. This performance of AFP LSE suggests that it can be effectively utilized for the annotation of hypothetical proteins. Furthermore, for an insightful understanding of CKSAAP representation-based classification of AFPs using the given dataset, we present a comparison of PCA and AFP LSE method. For visual assessments, the data were projected on 2 dimensions, utilizing the top 2 eigenvectors in case of PCA and 2 latent space in case of AFP LSE. The proposed nonlinear autoencoder based latent space encoding method presents superior learning capabilities as can be seen in figure 6C, and maps AFPs and non-AFPs in separate regions in contrast to the linear unsupervised subspace learning method of PCA as can be seen in figure 6A. The same eigenvector are then and the latent space from PCA and LSE are utilized obtained from the training are then utilized to project the test data. Differences in the mapping capabilities of AFPs can be observed for both PCA and AE-LSE method in figure 6B and 6D respectively. It can be observed at the bottom right of figure 6D that AE-LSE method forms cluster of AFP samples. This helps in understanding the working principle of proposed method and the motivation for development of non-linear autoencoder based learning for latent space. This brings us to the conclusion of the presentation. So herein we proposed a prediction method for antifreeze proteins in which each protein sequence was encoded using CKSAAP with k is equal to 8. For classification, we proposed novel machine learning based method for the AFP prediction, which uses an autoencoder for feature compression, and these reduced features are used to train the neural network based classifier. A comprehensive ablation study was performed for a better understanding of the effect of latent space variables, as well as the impact of training data distribution. And finally, widely used biostat nomenclatures were evaluated. We also compared the proposed nonlinear mapping method with the linear projection approach of PCA, which demonstrated superior classification capabilities of the proposed method. And finally, the results show that the method yields high values on the parameters of MCC, F1 score, and Youden index. Thank you.